and we'll get started with the recording going on. Just to give you an overview of what this project is all about, Aaron and Neon have partnered together. Aaron, as I said, is the Ecological Research as Education Network. And we're a group of ecologists, mostly working at smaller institutions to promote and support collaborative ecological research across multiple sites. So we use a common protocol to go out to all the different sites in our network that have volunteered to be part of a particular project, and people collect a common set of data. They share those data to allow for comparisons. We've been able to publish a number of peer-reviewed papers from these data. And it really gives a chance for undergraduates to learn about collaborative ecological science by engaging with it. We're very much of a grassroots network, so our project ideas come out of the different faculty participants in the network, and people kind of engage with the projects as they want to. We have over 300 members of ARIN, and if you're interested in joining ARIN, please feel free to visit our website at arinweb.org and join up. The National Ecological Observatory Network, or NEON, is kind of like ARIN's big brother, I would say. It collects the same type of data in the same type of spirit, where they're using the same protocol over a large network of 81 sites spread across the continent. They have some really impressive continental scale data products. And the projects we've developed for this flexible learning need that we all have during the pandemic really try to bring together that continental scale perspective with the perspectives that people working in the ARIN model can engage with, who are working more, let's say, at a regional scale or doing local data collection through hands-on activities that undergraduates can be part of. So that's kind of the idea. I want to remind everyone that we got our grant on July 17th, and we're all small college faculty working like all of you to respond to the pandemic. So we're kind of building this as we're implementing it. And we definitely like collaboration in developing our projects. And if you see ways that our methods could be better or our approach could be improved, please feel free to add that to the chat. So I'm going to go ahead now and introduce today's speaker and today's project. So let me take a moment and bring that information up here. All right. Dr. Allison Parker is an assistant professor in the Department of Biological Science and a member of the Environmental Science program at Northern Kentucky University. Her research interests focus on disease ecology in human-dominated systems, examining disease ecology from both ecological and social science perspectives. Her current research explores the relationships between human land use, mosquito abundance, and species diversity, and effective mosquito control strategies. So I'll turn it over to Allison, and she can go ahead and share her screen.
Awesome. Thank you for that introduction, Lori, and thank you all for coming today. Can everybody see my screen? Awesome. So today I'm going to go over the lab that I've been developing looking at mosquito surveys along anthropogenic or human impacted land use gradients. So the agenda for today, we're going to go over some background so everybody's on the same page about mosquitoes and why mosquitoes are important to study. Then we'll go over the research context in question. I'll then go over the core module, looking at mosquito distribution across human impacted land use gradients, which will include using neon data. Then I'll pause for some questions, and then I'll go over 2 additional modules looking at female oviposition or egg-laying choice assays and designing a mosquito control program. Then I'll go over some tips and tricks, including what is probably a burning question for all of you: how do you collect and rear mosquitoes at home, when to conduct this lab, and then we'll open it up for questions and answers. So why study mosquitoes? Globally, there's over 3,000 species of mosquitoes, and in the United States alone, we have 176 species. So we have a wide diversity of mosquito species, which makes it pretty cool to study in the United States. Mosquitoes are found in a wide range of habitats, which makes it really important for us right now when we're having students that might be face-to-face, but some might be at home. Students will be able to collect mosquitoes regardless of where they live, whether it's an urban area or if they live in a more agricultural area or even a suburban neighborhood. So that makes this project really flexible to meet your needs as an instructor and as for whatever your students might be facing and where they're actually living this semester. Another really cool part about this is that the mosquito species distribution changes across this human land use gradient. So we'll get different mosquito species depending on the disturbance of the land. And the other thing that makes mosquitoes really cool and somewhat easy to study is that they have a complex life cycle. So the female mosquitoes will lay her eggs on standing water or on the sides of a container holding standing water, or in something such as moist soil that will become flooded later. And we're going to be focusing more on these container breeding mosquitoes that lay their eggs in containers, because we can have a container outside that's easy to collect from. The larvae will then develop within the water, eating bacteria, fungi, decaying plant and animal detritus that falls into the water. After 4 instars, it'll pupate, go through metamorphosis, and then the adults emerge and begin their terrestrial life cycle. Males and females will both feed on sugar sources such as nectar, and only female mosquitoes blood feed because they need a protein-rich source to produce their eggs. So I just wanted to go over their life cycle to make sure that everybody's on the same page. So, like I said, mosquitoes lay their eggs, especially container breeding mosquitoes, on standing water or on the sides of the containers. So these are some pictures from some of my research from this summer, just to give you an idea of how easy it is to collect these eggs. In this first larger picture, all of these black little rice-like looking things are all egg rafts, and each egg raft was laid by 1 female mosquito. And that has about 100 to 300 eggs in it. So you could see there were many mosquitoes. I think in this particular day, we had over 100 egg rafts. The other strategy some mosquito species use is to lay their eggs on the sides of the containers. And you can see here all of these black areas, those are all hundreds of mosquito eggs. And later in this webinar, I'll talk about how we can collect them more easily by adding a substrate into the container instead of having them laying eggs on the side of the container. But being able to collect these eggs from a controlled container habitat makes this a project that students can easily do in their own backyards. It's also important to study mosquitoes because they spread a wide range of diseases. A lot of these diseases affect humans, but some diseases will also affect horses and dogs and birds. And so this is a cool tie-in because students that are in your classes that might not be as enthusiastic about ecology might like the idea of studying something that makes people or animals sick. And there's going to be an increased spread in mosquito-borne diseases in the future due to the new invasive mosquito species entering new regions and due to climate change. As you can see in this figure here, as we have climate change happening across our globe, there will be more suitable habitats and environments for these mosquitoes to live, and then the diseases they transmit will follow. So the main research question for our core module is, how does mosquito species distribution and diversity change across an anthropogenic land use gradient? And there's 2 predictions that I have. The first is that species diversity will be greater in undisturbed habitats. The second prediction is that invasive mosquito species, such as Aedes albopictus, the Asian tiger mosquito pictured here, will be more prevalent in highly altered urban environments. So we'll be looking at species diversity overall, but we can also look at species diversity specifically for invasive mosquito species. So the learning goals for all of the lab modules are to develop testable hypotheses for observational field studies, perform accurate data collection and recording of samples at field sites, even if that field site happens to be the windowsill outside your apartment, identify variables across field sites that may account for differences in our collected data, which will be really important if we have students who are doing this lab from home, analyze data collected by students and analyze data from NEON, interpret and draw conclusions from the analyzed data, and if you do one of the additional modules, students will practice communicating scientific topics to the public. So what will be provided to you as the instructor? You will be provided background PowerPoints and background reading in the lab manuals for all the modules. So you can either introduce the topic using a PowerPoint, or you can have students read about it in their lab manuals. In the lab manuals, you will get an instructor version, which will include notes and tips to help you along the way. And then there'll be a student version for students to enter in their answers to each of the questions that are throughout the lab. You will also get instructions for using NEON data, and the NEON data we'll be using for this project is called Mosquitoes Sampled from CO2 Traps. You will get data entry templates and data analysis templates to aid your students in collecting their data and aid them in analysis. Within each lab manual, there will be driving questions to work through the scientific method and engage students with the material. And there'll also be reflection questions provided. For each module, I will also provide primary literature so that you can have your students read primary literature and have journal discussions if you like. And then the goal of this is to make a data repository from the instructors, from you, for future semesters. So after you collect your data, you will send it to me, and then I will make a repository so that in future semesters, we'll have data across a wide range of land use types. So first, let's talk about the core module, which is looking at mosquito distribution across land use gradients. So in this module, students are going to collect and rear mosquitoes either in the lab if you're face-to-face, or they'll collect and rear mosquitoes at their home. And later in the webinar, I will go over all the steps on how to do this. It's not as hard as it sounds. Students will also identify adult mosquitoes to species. And the reason that we're going to do this is because NEON protocol, they collect adult mosquitoes and they identify them down to species. So to be able to use your students' data and the NEON data, we want to be able to do that. I'll provide taxonomic keys with this module to help you. And I also created an iNaturalist project where your students can post pictures of their adult mosquitoes for help with identification. The nice thing about identifying adult mosquitoes is, for the most part, you can see a lot of their distinguishing characteristics without the use of a dissecting microscope. And there's also apps that students can download on their smartphone that allow for magnification just through their smartphone camera. So there's lots of different ways that students can magnify something if they don't have access to a dissecting camera. Students are then going to use canopy cover and impervious surface data to determine the level of human impact. So whether it's at a field site by your campus or if it's in their own backyard. And to do this, they'll use Google Earth and the Multi-Resolution Land Characteristics Consortium webpage. So this will help us get a quantitative view of how disturbed their particular habitat is. Students will also use NEON data. And the really cool thing about the NEON data is that we have mosquito collection across the 20 ecoclimate domains, which you can see on this map here. So if, for example, if you're collecting mosquitoes, your students are collecting mosquitoes in New York, you can compare your students' data to domain 1, the Northeast, to see what similarities and differences there may be. All of the habitats for the NEON data are undisturbed habitats. They may be forested habitats, they may be grassland, It just depends on the particular ecoclimate domain, but your students will have wherever they're living to compare to the undisturbed neon habitat sampling. Another really great thing about the neon data is that they started collecting it in 2014. So if your students are interested in looking at how mosquito species diversity changes over time instead of across locations, that's also something they can easily look at because that data is available to you. Students will then analyze differences in species diversity. So they'll analyze their own data within their class, then they will do some neon data analyses, and they'll do some diversity indices and rarefaction curves. And then what is really great about looking at these two different levels of data collection is it leads for really great discussion on sample size, which I think is really important in ecology and sometimes is difficult for students to grasp. But using these two different data sets, one that they and their classmates have collected and the neon data, they can really start thinking about what sample size means and how that might affect the results of their analyses. And then, like I said earlier, we'll develop a repository for data across a wide land use gradient. So in the future, if your students are interested in comparing, say, a suburban neighborhood to an urban area, they'll have that data available to them. But right now, we only have undisturbed habitat to compare to. So I wanted to go over what the neon data looks like because I know it might seem a little overwhelming to think that your students are going to actually access this data. So I'm going to show you this Excel sheet first, and this is what the mosquito identification data looks like. So you can see here, it has your domain ID, which this is from domain 1, which is the Northeast domain. You have your plot ID, and that will become important when I show you the next Excel sheet. You have your collection date and other important information. What's really nice about this data is they have the individual count, which makes putting this information into like a Shannon diversity index very easy because they give you the number that they counted, then they have the genus, and then they have the species. And so the students can go across and figure out what the taxons are and then the individual counts. So that makes it very easy for students to actually access and use this data. If you're interested in what does the plot ID of BART 056 look like, there's actually Excel sheets that will show you that as well. So this will be downloaded within the same file, and I have instructions on what files you're supposed to open. So you would just go here, you see your plot ID is BART 056, which is what all of these mosquitoes were collected from. And you can see that this is a deciduous forest. It gives you other information like the coordinates and collection dates and things like that. It also gives you night and day because in neon they do sampling at night and then they do it the next day as well. So if you wanted your students to compare species diversity between the night and the day, you can do something like that as well. So this data is set up really well for students to learn what large data sets look like. So I'm going to pause here and see if we have any questions in the chat about this core module. Okay, so there was a question I saw. Does the CO2 trap data from local county mosquito commissions? No. So for those of you who aren't up to date on how NEON does their mosquito sampling, they put out CDC CO2 traps, which collect adult mosquitoes, and NEON themselves puts those out and collects them at their sites. They do send out the mosquitoes to independent groups to do the identification, but NEON themselves are the ones who are doing the collection from the CO2 traps.
Any other questions people wish to add to the chat at this time? There's another one coming through. So
somebody asked about EEE, which is Eastern equine encephalitis, especially if you live in the Massachusetts area. There's been some cases there already this summer, and I'm going to talk later what to do if students don't want to actually collect mosquitoes and why it's actually okay to collect mosquitoes for the most part, the way that we're going to do it. But I will come back to this later.
And one other about DNA extraction of the neon mosquitoes.
Yeah, so there's two other data sets for mosquitoes in neon, that Neon does. They do DNA extractions for the mosquitoes to get better species identification, and they also do pathogen testing. Because this was supposed to be geared more towards general ecology, I didn't make any additional modules with that, but if there is interest in using that data, I'm more than happy to develop modules that look more at pathogen transmission or the DNA extraction.
And I see Megan Jones, who is our contact person, one of our two contact people we work with at Neon, has put up a nice link to some of those data products. Anything else people want to add before we continue on? A question about doing this in areas where malaria and dengue are endemic.
Okay, so I know somebody asked about Eastern equine encephalitis, so I'll just touch on this now. We are going to be collecting mosquito eggs for this project. And so we're not going to be collecting females that might have blood fed on an infectious blood meal. So the students will rear their mosquitoes from eggs to adulthood. And so there's a very, very, very small chance that a student would be able to get a mosquito-borne disease from their collections.
Sounds good.
Okay, so now I'm going to talk about two additional modules that can be added onto the core module or just done separately if you don't want to do the core module. The idea is you can pick and choose what you want to do for whatever best suits your class. So the first module is looking at the question, do female mosquitoes prefer to oviposit or lay eggs in certain container habitats over others? So this is a really great module, especially for introductory ecology classes or classes where students are really learning how to use the scientific method and develop their own hypotheses. So students will be given a list of independent variables, some of which you see on this slide, and they're going to be able to develop their own hypotheses and predictions and develop their own experimental design to test this question. And so students can test things such as water volume or surface area. It can be as simple, especially if your students are doing this at home and have limited resources, you could do container color. So they could put out one red plastic cup and one blue plastic cup and see if females have a preference. And then students would just do a t-test. So there's lots of easy ways that students can do this even at home. You can look at nutrient levels. So you can add grass clippings to one and just have plain water. You can add fish food or some other dried pet food to one and nothing to the other. There's lots of different ways to affect the nutrient level in the water. And you could even do height of the container, where a student would put one container on a windowsill and the other on the ground by the windowsill. So there's lots of easy manipulations you can do to these containers that students can test on their own. So here's a picture of what some of my research students looked at this summer. They were really interested in if you change the volume of the water, but the containers are identical to each other, do female mosquitoes have an oviposition preference? So this is just something easy even my research students are currently working on. One of the great things, other than the fact that students can develop their own hypotheses and develop their own predictions and have some manipulation of an experiment, even if they're at home, is that this module is easily integrated into the core module. So the only difference or the only addition to the core module, if you do this, is that students would then count their mosquito eggs. Because these containers, as you can see in this picture, have to be so close to each other, we can still use what is collected, the adults that we get from these eggs, as our data for the core module, comparing it to our NEON data. So this is easily integrated into the core module if you would like to do both, especially if you want your students to do a more hypothesis-driven experiment that they're actually driving the hypotheses and the predictions. So the second additional module, and this is kind of coming from my social science background, is for students to design a mosquito control program. And the students can pick, and you could either have them do this individually or in groups. They can either pick a mosquito species, so say that there was a large number of one particular species that the students found in their containers. Maybe they're really interested in exploring that species some more. Even if it's just a pestiferous species, it's still worth trying to control. Or if they're interested in a particular mosquito-borne disease, they can pick that and research that instead. Aspects of their program that they have to think about is the location of their program. So if students are really interested in looking at malaria, but they want to put a malaria control program in Kentucky, that's not going to work because we've eradicated malaria in the United States. So they have to think about where is the location going to be. They have to think about how they're going to implement control strategies and what are those control strategies going to be. And you'll have a PowerPoint that will go over various types of control strategies that can be implemented for mosquito control. Students then will also have to think about how does culture and personal values of members of their community affect their control program. Some communities might be very opposed to the use of insecticides. So what are you going to do as the mosquito control expert to have your community get on board with your design? They'll have to design some sort of educational outreach to the community to let them know about this control program that they're going to be implementing. And the really cool thing about this is that they can include the results from their own experiment into this control program. So if you're collecting mosquitoes, you can have the students actually design the control program for their local area, which I think is something that's really cool because a lot of times we collect data in labs, but students don't really get to see what we do with the data. So this is a way to show in real world what mosquito ecologists do with their data. We use this data to design control programs. Then they could present the programs to the class. And this is a great way for students to think about how to actually communicate scientific topics to the general public. If there's jargon or if there's complicated figures, how do you break those down so people that don't have a science background can actually understand it? And I think that's something that's really important now more than ever. So those are the two additional modules that I have already, and I'm more than happy to hear ideas for other modules that people are interested in. So now I'm going to go over tips and tricks, including how to collect and rear mosquitoes in your own home. So these are the materials that you need to collect mosquitoes. You need some sort of plastic containers, like you can see in this picture, just some sort of soup container. Any plastic container will work. You need some water, you need popsicle sticks, a paintbrush or an old toothbrush would even work. A dissecting microscope is great, but if you are at home, you can use a hand lens. There's actually clip-on microscopes that you can attach to your smartphone, or you could use a magnification app that you can download for free on your smartphone. To rear our mosquitoes, we're going to feed them fish food, or really any hard dried animal food works. Dog food, cat food, rabbit food, but fish food works really well. And then you'll need an emergence trap. And I'll show you what that looks like in a few slides. And there's instructions for constructing those at home included in the lab manual. And you just use things that most people will have around their house or are very inexpensive to purchase. But you can also order emergence traps from bioequip.com if you want. So how do you collect mosquito eggs? All that you need to do is put out a container of water with a popsicle stick in it. You leave that for about 5 to 7 days, and you should get some mosquito eggs. So here is the popsicle stick from this container this is outside where I live in downtown Cincinnati. And so all of these little black specks are mosquito eggs. And I have not zoomed in on this. This is just taking a picture with my smartphone. So if you decide to do the oviposition choice assay, even just zooming in a little bit on your smartphone you can count most of the eggs that are laid. Unless you get hundreds of eggs and they're all very close to each other, then you might need to actually use something better than just your camera. The other types of eggs that are laid are egg rafts, which I showed you the picture of earlier in the talk. And there's 4 egg rafts in this container. 2 are just next to each other. Just for scale, this is what an egg raft looks like on the tip of my finger. There is about 100 to 300 eggs laid here. To collect these, all you do is you scoop them up with a paintbrush, and they usually just stick on the tip of the paintbrush, and then you can move them into a new container of water. So it's very easy to collect these eggs. So you just leave out this water-filled container for 5 to 7 days, and then you bring it inside. Or if you want to, your students can actually leave it outside as long as the weather is not terrible. So how do you rear the mosquitoes? You will submerge the eggs that are on the popsicle stick, so just add some more water to your container. Those eggs and the egg rafts will hatch within a few days. It could take anywhere from 1 to 4 days since once they're laid for the eggs to hatch. The hatching larvae are very small, especially right after they hatch. Their exoskeleton hasn't hardened, so they might be hard to see. So just tell students that they're probably in there and not to worry. Each day you'll feed them a small amount of fish food or whatever hard pet food students have access to. Once one pupae appears, you need to ensure that there's enough food for the remaining larvae, and then you attach your emergence trap. So this is what the emergence trap looks like. So this was the initial container that I had collected my eggs in. And what you do is you glue 2 tops together from 2 containers that have the same size opening. And then you use a cut bottle and just cut holes in the middle and put that bottle in there. You glue this all together. And what will happen is your pupae, once they emerge as adults, the adults will naturally fly up. They will fly through the opening of that bottle cap, and they will be in this upper chamber. And then you cut a hole in the top with some mesh so that there's enough oxygen for the individuals. Once all of your larvae have pupated, you could place this, once all the pupae have emerged as adults, you can place this into the freezer for 30 minutes to an hour, and it will anesthetize the adults. And then students can take out their emergence trap, pop off the top that the adults are in, and put just a normal lid on top and put that back into the freezer. The great thing about that is you can leave the adults in the freezer until you're able to get to identification. So if you're trying to get all of your field sampling done in the beginning of the semester because you're worried you're going to get transitioned to going all online, or because you live in a more temperate region, so your field season time is shorter, you can actually leave these and do the adult identification later in the semester. So a lot of people have asked me, when should I conduct this lab? The best time to collect mosquitoes is immediately at the beginning of the fall semester. Even if you're planning on doing this lab later in the semester, you can have students put out their containers, collect the eggs, rear them to adulthood, and like I said, put the adults in the freezer until you can go more into depth for this lab. This is really important if you live in more temperate regions. If you live in more subtropical regions, you can do this lab anytime throughout the semester, but it's still best to do it now because there's more mosquitoes out now than will be later in the semester. I know some people were asking about, oh, I'm not sure if my students feel comfortable collecting mosquitoes, and I totally understand that. And so what you can do if you don't feel comfortable or your students don't feel comfortable collecting the mosquitoes, you can actually just conduct the core module and only have students look at neon data. And so they can look at the data across different ecoclimate domains so they can see how species diversity might change between the Northeast and a different domain that they're interested in. Or you can have students look at changes in species diversity over time, because like I said, there is data from starting at 2014 from some sites, but all the sites have data from 2016 on. And so if you don't feel comfortable collecting the mosquitoes, this is another option for you. And you can still do the Design the Mosquito Control project as well. So I wanted to make sure I kept it brief so that there were enough time for questions. So with that, I will open it up to any questions anybody has.
So there are a couple. We've got somebody asking about the temperature limits on mosquito sampling. For example, here in Anchorage, Alaska, the first freeze tends to happen around the first week of our fall semester. So I guess any potential for starting later than that? Or yeah, beyond that first week?
Um, if you are in Alaska, I would suggest getting this out as soon as possible. After the first freeze, it might be a little bit more tricky to get mosquitoes. There might be a few lingering around. Those last females might want to lay their eggs. But yeah, Alaska might be a little trickier to do. It just kind of depends on when that freeze happens.
The next one is, are container mosquitoes the same kind that occur in pitcher plants?
Typically, the pitcher plant mosquitoes are more specialized to those particular pitcher plants, but sometimes container breeding mosquitoes will lay their eggs in pitcher plants. The natural containers that a lot of these mosquitoes would lay their eggs in are tree holes. So when branches of trees come together and form a depression, that was what their more natural container habitat would have been.
How early in the calendar year would you recommend starting in Kentucky?
So I would recommend, if you're going to start this fall, I would recommend putting out your egg collection containers as soon as possible. So whenever your semester starts would be the best. In Kentucky, we'll have mosquitoes probably through October unless there's an early freeze, but they'll start to dwindle down in September and into October. So the sooner you can get them out in August, the better.
Can you use any water for rearing, or do they need spring water versus tap water?
That's a great question. So you can use any water. If you're using tap water, in the lab manual, it will suggest to leave the water out for 24 hours so that some of that chlorine can dissipate from the water, but they don't need any special treatment to the water or anything like that.
What is the phone magnifier app that you recommend?
Let me look up what it's called on my phone. There's a few different ones, and it depends on if you have an iPhone or an Android. The one that I've been testing out and piloting to see if it would work for this lab is called Magnifying Glass, and it's free. So I'll have a list of apps for students to pick from depending on their phone, and they'll all be free options.
We have one person showing some appreciation for the Erin and Neon projects. I'm really glad we can offer these to you, and I want to especially thank the presenters we've had so far, including Allison today, because they are doing a huge amount of work to get these things ready for everyone. But it's really exciting that we can work together to address some of these huge challenges that we have this semester. So excellent. Someone asked, have you developed any modules for using the CDC protocol for looking at the evolution of insecticide resistance? Alternatively, are there data from a variety of locations that one could use?
So I haven't thought about doing that. That is something that could be developed. I would just have to look into what the safety requirements would be for students working with some of these insecticides. I don't know if it would be great for students to do this at home, but definitely in a face-to-face ecology lab, that would definitely be something that could be developed in the future. There is data from a variety of locations. I'm just not sure if it's open access or not, but I can definitely look into that. I'll make a note.
Let's see, and actually the question we had about timing was in relation to the spring semester. So if you were going to do this project in the spring semester, when would you recommend starting the collection in Kentucky?
So in the spring semester, it kind of depends on precipitation and temperature. You could probably start getting mosquitoes as early as mid-April. If you're further south in Kentucky, you might have a decent amount of mosquitoes mid-April. It just kind of depends on the weather and when the thaws happen and things like that.
So here's one about student collected data from the core module. This says student collected data from the core module cannot be directly compared to NEON data. Is that correct? How do you envision students in different situations, for example, an urban apartment, suburban park, standardize their data to compare abundance, diversity, and so on with each other's sites?
So that'll be part of their data entry template that they'll have. And so they'll use Google Earth and the MCRL website to basically, it'll give them a percentage of canopy cover and impervious surface. And so then we'll have that gradient. And one thing that I will suggest that will be suggested in the lab manual is that you split your students into two groups of either impervious surface or canopy cover, depending on the locations your students might be collecting at. And then this way you can have a more disturbed and less disturbed habitat. And then you can do a Shannon diversity index between those two basically gradients of more disturbed and less disturbed. The idea is then you would also do it for, you could pick 2 neon locations. What is going to happen then is there will be an additional analysis where students will take their data from one of those two, you could do say the more disturbed, more impervious surfaces, and compare it to a neon site, with the idea that students will then see that the number of mosquitoes they collected is so much smaller than the neon data that it makes it really hard to compare those two. And that was what will lead to that discussion about sample size. So they will be able to compare it, and I'll have instructions in the lab manual about how to go about doing that.
That's a great point, that we can teach a lot about the scientific method and kind of scientific practice. We really did want to take advantage of the neon data as a way to think about data science and just data management skills. So yeah, that will be an exciting aspect of this project. Another question here, did you make a tree slurry for your collection? I had read somewhere to put some leaves and other debris in the water before collection to help attract mosquitoes.
So I did not. I just put some tap water I'd let sit for 24 hours and put it outside. And things fell into the water, and that's why it takes about a week for the mosquitoes to lay the eggs. If you want, that's something you can have your students do. There will be a line in the data entry template that'll say students put grass clippings or oak leaves or whatever, but they do not need to do that. It does make it more attractive, and you'll get eggs more quickly and more eggs, honestly, but it's not required.
Actually, I'll jump in with my own question. I was wondering if you needed to note or standardize the surface area of the object you put in for egg collection. Like, should it be a popsicle stick of a certain dimensions, or should you take note of the dimensions?
You can, but for the popsicle stick, it's really just giving the mosquitoes a substrate that they will prefer to oviposit on instead of the sides of the container, just to make it easier to actually see the eggs. So you don't need to standardize across that, as long as it's some wood or kind of fake wood substrate, that's fine.
Someone asks, if we can't do the lab this semester, but want to use it in a future fall semester, do we need to notify you?
You don't need to notify me, but if you want to see the data that we've collected this year, and then just send me an email and I can share it with you if it's not available online yet. And if you want to send me your data after you do it, even if it's next year, that would be great.
Also, I'll just jump in. I don't want to at all speak for Allison, but we envision these 4 projects we're developing quickly in response to the pandemic as kind of pilot errand projects. And errand projects often run for 1 to 2 or even more years. Sometimes they'll run for several years. But we often make the decision about how long-term the projects will be after doing an initial round of data collection and just seeing how the technique's going, and does it look like a promising study? So this is certainly something I could imagine if Allison wanted to continue, Erin would be excited to support it. But I do feel we're kind of in a pilot phase realistically at this stage, and we're going to see how it works. Someone asked, what do you recommend for permanent preservation of the specimens? Pinning on points or I'm sorry, in minutions and double mounts, alcohol, glue to glass slides. I would like to donate the identified specimens to a natural history collection.
So just for ease, I like to pin my mosquitoes on points. That's the easiest to work with personally. I do not suggest putting them in alcohol because one of the main ways that we identify adult mosquitoes is by their scales. And if you put them in alcohol, they'll fall off. So we won't be able to see it. So I think for students, especially if they're not in an entomology class and that's not a skill that they're really focusing on learning, I would not do minute pinning if I could avoid it, but pinning on point is totally fine.
Someone asks, when will the resources be available to instructors? We're trying to get everything posted to our website, at least in a usable form. Once again, we're imagining these modules being improved over time, but we'd like to see everything posted by August 21st, so a week from Friday. And we know that that will be very close to the start of the semester, but we're hoping that these webinars will give you enough of a sense of how things are structured that you'd be able to plan for it in your syllabi. Let's see. And Tim has put a link to our flexible projects. I think that's all we've got in terms of specific questions. Some people are saying that they may not have a chance to try it until next fall, but that they're excited about that. So great. Well, if no one else has any specific questions, Allison, thank you very much. And I'll remind you that we have another module tomorrow that will be presented this time at 10:30 in the morning. And this will be on lichens, looking at lichen diversity and abundance across landscapes. Once again, with the flexibility and social distancing built into the projects as best we can. We are going to save the chat, so if you have any last minute comments to Allison, please go ahead and add them in there. And there is another question where someone says, back to students, how students use data. Do they compare egg data or raised adult data?
So students, if you do the oviposition choice assay, they will be comparing egg data because not all the eggs hatch. And so we want to have an accurate count of what the females are actually choosing. But for the core module, we'll be looking at adult data.
We are going to have this webinar available on the website that we've linked in the chat on the Erin website. We will post the webinar for future viewing. And yeah, we're just, I'm hoping to have all of those posted by the end of the week by Friday so that people can take advantage of that. So, all right. Well, thanks again to Allison. Please give her a virtual, oh, actually, I'm sorry. Megan Jones was saying, yeah, no, I'm sorry. She was just sending me a message. But thank you very much to Allison. Let's give her a virtual round of applause. Sorry, distracted by the chat.
Thank you all for coming.
Yes,
and if
thank you very much for coming.
Questions, feel free to send me an email.
Yes, indeed. And see some of you maybe tomorrow morning at 10:30.
Do you want to stop the recording? All right.
Yep. Thank you for the reminder.